Format, ~950 words conversational narrative. Target, AI tech audiences, ~5-minute watch time. There's a lot of hype around AI in healthcare, robots reading radiology films, chatbots triaging patients, all the big flashy stuff. But this week, the papers that actually landed tell a very different story. The most impactful AI for healthcare right now isn't necessarily building new sensors or new hardware. It's finding signals in data we're already collecting and extracting meaning nobody knew was there. Today we're covering 5 papers that range from pancreatic cancer screening to drug discovery to IBD diagnosis, and one important warning about structured data that the LLM crowd needs to hear. Let's get into it. First up, a paper out this week on AI-powered pancreatic cancer screening that genuinely made me stop and read it twice. Pancreatic cancer has a 5-year survival rate around 12%. Part of the reason is brutal, there's no standard screening test. By the time symptoms show up, it's usually too late. But what if you could flag high-risk patients years earlier, using data that's already sitting in someone's medical record? That's exactly what this team did. They trained a transformer with multi-head attention on routine blood tests and clinical histories from over 6,000 patients and 177,000 controls. Not specialized biopsies, not expensive imaging, just the ordinary blood panels and visit records that healthcare systems already collect by default. The result, an AUC of 0.837 at a 1-year lead time. That means the model can predict which patients are at elevated risk a full year before conventional diagnosis would catch it. The authors call it the first population-level digital enrichment tool for pancreatic cancer screening, and honestly, that framing is right. This isn't about replacing doctors, it's about telling a doctor, hey, this patient in your normal risk cohort might need a closer look. Second paper is about drug synergy, specifically, predicting when 2 drugs together are more effective than either alone, in situations the model hasn't seen before. This is a hard problem. Drug combinations are combinatorial, there are millions of possibilities, and you can't test them all in a lab. Machine learning helps, but most models fail when they encounter drug pairs from new, unseen chemical families. That's called out-of-distribution generalization, and it's the difference between a model that works on paper and one that works in the real world. Enter OOD-GraphLLM. This is the first framework that jointly optimizes molecular graph representations and biomedical semantic representations. Think of it as training the model to understand both the molecular structure of a drug and the biological context it operates in, simultaneously. By aligning those 2 representations, the model can predict synergies for drug combinations it's never seen before, because it understands the underlying biology, not just memorized chemistry. This is the kind of architecture that might actually accelerate early-stage drug discovery, especially for rare diseases where you don't have mountains of existing data. Third paper touches on something called iLoRA, and I'll be honest, the name is dense, but the idea is elegant. LoRA, or low-rank adaptation, has become the go-to method for fine-tuning large models efficiently. Instead of updating every parameter, you inject small trainable modules. Standard stuff by now. What iLoRA does is different. It infers a latent interaction graph, a hidden network of relationships between input features, and then uses that graph to condition how the LoRA updates themselves are generated. Think of it this way, standard LoRA gives every input the same adaptation strategy. iLoRA learns which inputs interact with each other and tailors the adaptation accordingly. That Bayesian component means it also quantifies uncertainty, it knows when it doesn't know. The team tested this on microbiome diagnosis for inflammatory bowel disease. Your gut microbiome is a complex ecosystem, the relationships between bacteria matter more than which ones are present. iLoRA naturally captures that because it's literally designed to learn interaction graphs. It's a reminder that sometimes the architectural innovation matters more than the benchmark number. And now the warning I promised. We had a paper this week called MedCase-Structured that built a benchmark pipeline using FHIR, that's the standard format for electronic health records. They compared how LLMs perform on structured clinical data versus plain text descriptions of the same cases. The finding was unambiguous, models performed worse on structured inputs than plain text. Significantly worse. This matters because real healthcare data lives in structured formats, databases, JSON payloads, ontologies. Most LLM demos use clean narrative text that's been curated. The messy, structured reality of clinical data? Models struggle with it. If you're building or buying a healthcare AI product and it's only been tested on narrative text, you're going to get a nasty surprise when it hits real structured data. This paper is a reality check the field needed. 2 quick honorable mentions before we wrap. LUMI is a mental health preference tuning dataset derived from Reddit, think RLHF for therapeutic dialogue. And VisAnomReasoner is a VLM-based medical monitoring system that improved anomaly detection precision by over 21 percentage points. Both worth a look if those areas interest you, and links are in the description. Here's the thread running through everything this week. The most clinically impactful AI right now is the stuff that extracts latent signal from data healthcare systems already collect. It's not waiting for new hardware or new data types. It's saying your routine blood work actually contains a pancreatic cancer signal. It's saying the standard way you structure clinical records is making your LLMs worse. It's saying the relationships in your gut microbiome are a graph we can learn. That's the frontier. Not building better scanners.